All right. Uh, Gives me great pleasure to welcome Carl Veller to UCLA to come and talk to us. Carl originally studied maths and economics in Cape Town, and then he started a PhD in economics at Harvard. Then he saw the light and converted to evolutionary biology, which he completed his PhD in under the supervision of Martin Nowak. Then he did his postdoc with Graham Coop and Chuck Langley at UC Davis. And that's when I first became aware of Carl. He wrote a couple of really impressive papers using sort of rigorous approaches from population genetics and econometrics and applying them to family-based methods, which is my own area of research. And I was actually sent both of Carl's papers to review. I hope he's forgiven me. And I learned a lot from them. They were very interesting, and I'm sure you'll learn a lot also from Carl's talk today. So please join me in welcoming.
Okay, thanks very much, Alex, for the introduction. Yeah, so I'm going to talk today about stabilizing selection, which in the context of quantitative trait is simply selection for an optimal value of that trait, where larger deviations from that optimal trait value are penalized more severely by selection. Now, stabilizing selection is probably the most common form of selection in natural populations. I say probably, there's not an enormous amount of direct evidence for stabilizing selection, but what I mean is that sort of intuitively we know it's true, right? We think of features of birds' wings as having been perfected for properties of their flight, and presumably those features remain under stabilizing selection for that purpose. There's a lot of circumstantial evidence that stabilizing selection must be the most common form. Paleontologists will talk of, enormous periods of phenotypic stasis in the fossil record. In terms of actual direct evidence, it's quite hard to get because you need measurements of fitness for a range of individuals, and you need measurements of the trait that you believe to be under stabilizing selection. And in fact, it has been shown, in a number of species, including in humans, that stabilizing selection does act on a lot of quantitative traits. This is an example for human height where these authors used lifetime reproductive success, the number of offspring one has in one's lifetime, as a measure of fitness, and showed that individuals that are very tall, very short, tend to have slightly fewer offspring than individuals with an intermediate height, both in males and females. So even for like a classic quantitative trait like height, there appears to be, in this case, weak stabilizing selection. Okay, so stabilizing selection is surely extremely common, but it's probably, seems like quite a boring thing to study. And that's because its macroscopic dynamics are pretty straightforward, right? We place a trait under stabilizing selection for some optimal value. The mean value in the population will move to that optimum, and the variance of the trait will contract under stabilizing selection. Okay, so that's a pretty boring macroscopic picture. It turns out, however, that if you look under the hood, the microscopic genetic dynamics that underlie this process are extremely rich and sometimes quite counterintuitive. So for example, this reduction in the genetic variance of a trait under stabilizing selection happens via two distinct mechanisms. Okay, the first mechanism is that by selecting for compensating combinations of trait increasing and trait decreasing alleles, stabilizing selection very rapidly generates correlations, linkage disequilibria, between alleles of opposite effect on the trait. This has come to be known as the Bulmer effect. And it very rapidly reduces genetic variance in the trait. The second effect is that stabilizing selection generates quite interesting allele frequency dynamics at individual loci. Now, to understand these, imagine that we parameterize stabilizing selection as a Gaussian fitness function with a certain width, Vs, right? So larger values of Vs correspond to weaker stabilizing selection, a broader fitness function. We imagine that the phenotype is centered on the optimum, but that there's some variance in the population, Vp, and we focus on a particular locus contributing variance to this trait. Okay, and at this locus, there are two alleles. Allele big A has frequency P, allele little a has frequency 1 minus P. The effect of allele big A is to increase the trait value on average by an amount alpha, and that's versus relative to the effect of the alternative allele little a, which we can think of as a null allele with no effect on the trait. Okay, and under this model, Alan Robertson showed quite a long time ago that the frequency dynamics expected at the locus are given by this formula. So if you look at that term in green, it shows that if the frequency of big A is less than 1/2, if it's the minor allele at its locus, then P is expected to decrease. That allele is expected to be selected against. So stabilizing selection selects against rare alleles. And the intuition for this effect is quite straightforward. Okay? If we think of the same locus, the mean effect of an allele at this locus is P times alpha. Okay? For the mean phenotype to be at its optimum, which I've arbitrarily coded as 0, that means that the mean effect of the rest of the genome must be minus P alpha to compensate for the effect at our locus. So the mean phenotype of allele big A, right, is its effect on the trait, alpha, together with the genome-wide mean effect on the trait minus P alpha. In other words, 1 minus P alpha. While the mean phenotype inhabited by the little a allele is 0, its effect on the trait, together with the mean effect of the rest of the genome minus P alpha. And you can see that precisely when P is less than 1/2, when big A is the minor allele, the mean phenotype that it inhabits is further away from the optimal value of 0 than the mean phenotype inhabited by little a. So to put that into words, the rest of the genome must adapt to the common allele at our locus, and that leaves the rare allele at our locus maladapted to the rest of the genome. Okay, the frequency dynamics that stabilizing selection induces at individual loci are relatively slow. And so we have this kind of separation of timescales where the reduction in variance of a trait placed under stabilizing selection is initially entirely due to the Bulmer effect, to the linkage disequilibria generated by stabilizing selection. And then on a much slower timescale, you have selection against rare alleles at individual loci, eroding heterozygosity at those loci and thus reducing variance in the trait. And in fact, this separation of timescales can be used to show that the linkage disequilibrium that stabilizing selection very rapidly generates, in fact, influences the allele frequency dynamics. Okay, and so to see this, remember that the big A allele that had a plus effect on the trait, under the Bulmer effect, is expected to come into a correlation with minus alleles elsewhere in the genome. And those minus alleles are going to partially mask the trait increasing effect of big A, and that's going to slow down the frequency dynamics at its locus. Again, a graduate student of my group, Sharif Negam, and I have shown that this linkage disequilibrium effect can be taken into account in the classical formula for allele frequency change under stabilizing selection in a way that substantially improves prediction of trajectories of allele frequency dynamics, at least in simulations. Okay, but to an evolutionary biologist, the interesting question should be, does stabilizing selection have interesting evolutionary consequences? Okay, and that sounds a little bit like an oxymoron, right? The characteristic macroscopic effect of stabilizing selection is phenotypic stasis, and that's pretty much the opposite of an interesting evolutionary consequence. Okay, but as I've shown, if you look behind this phenotypic stasis, you see an interesting, quite rich genetic churn. And so the question really is, does that genetic churn under stabilizing selection lead to any interesting evolutionary consequences? So I'm going to give 2 examples where I think it does. Okay, the first relates to hybridization and speciation. And this is work I've done with a colleague at UChicago, Yuval Simons. And the second relates to transitions between sex-determining mechanisms. And this is work of a collaborator and colleague at UChicago, Pavitra Muralidhar. Now, it's going to turn out that for both of the phenomena that I discuss, they're going to be driven certainly not by the macroscopic effects of stabilizing selection, because as we've discussed, those are pretty boring. But they're also not going to be driven by the microscopic genetic effects of stabilizing selection. That is, they're not going to be driven by these dynamics of allele frequencies at individual loci. Instead, both of these phenomena are going to be driven by a kind of intermediate scale, a chromosome scale dynamics that stabilizing selection generates. And so in between talking about these 2 phenomena, I'm going to talk about some theoretical work that I've been doing with Pavitra to characterize theoretically these mesoscopic dynamics under stabilizing selection. And we'll show that they're kind of qualitatively different, actually, from both the macroscopic and the microscopic dynamics. Okay, so on to the first example to do with hybridization and speciation. So it's now widely appreciated that many species, maybe most species, certainly our own, have histories of hybridization and subsequent genetic introgression. Okay, and so for these species to remain genetically distinct, there must be some mechanism, right, by which introgressed ancestry is purged, right? That's how these species will remain genetically distinct in the face of this gene flow. And so the question is, what is the mechanism by which introgressed ancestry tends to be deleterious? Now, the classical explanation is Dobzhansky-Müller incompatibilities, alleles that substitute in the separate parent populations come together in a hybrid individual and are found to be incompatible. They've never experienced each other, these alleles in the parent population, so they've come together for the first time in the hybrid, and they reduce the hybrid's fitness through some kind of negative epistasis. Another possibility is that alleles carried by the donor species, the introgressing species, are adapted to the donor species ecology, right? And so if the donor species ecology differs from the recipient species ecology, then those alleles, when they enter the recipient species, might be maladapted to the recipient species ecology and cause a reduction in fitness in hybrids. A more recent explanation that's been argued to be particularly pertinent to Neanderthal human introgression is the possibility that the donor species has a lower effective population size than the recipient species. And this has been argued to be pertinent to Neanderthal human introgression and other cases because population genomic studies over the past decade and a bit have suggested that the deleterious effect of introgressed ancestry shows signals genome-wide. So there seems to be genome-wide selection against introgressed ancestry in many cases. For example, it's been estimated that Neanderthal variants were deleterious in humans at more than 1,000 loci throughout our genome. Okay, what I'm going to argue is that another possibility for selection against introgression that should show a similar genome-wide effect is simply stabilizing selection on quantitative traits. Okay, and so to argue for this, I'm going to consider the simplest possible model. I'm going to imagine an ancestral population up here in gray. It's under stabilizing selection of a certain strength for an optimal phenotype of 0. So I'm considering a single trait under stabilizing selection. This ancestral population then splits into two descendant populations that remain under exactly the same regime of stabilizing selection for the same optimal value and of the same strength. Those populations have some time to diverge from each other, after which they mix in unequal proportions to form an admixed population, and that admixed population is also going to be under the same regime of stabilizing selection. Okay, and it's important that these two populations are admixing unequally. So in this case, the yellow population is contributing a proportion theta of ancestry to the admixed population. I'm going to assume that theta is small. Okay, and I'm going to call the yellow population 2 ancestry the minor parent ancestry, and I'm going to say that it is introgressing into the major parent population, population 1. And the question is, what happens to the minor parent ancestry in the admixed population? And it's reasonable to think that the answer is nothing, right? The two parent populations I've assumed to be demographically identical. They're under the same regime of stabilizing selection. So they're centered on the same optimal trait value with the same genetic variance. They have the same average fitness. And so if I now create an admixed population of those two parent populations, because the parent populations are macroscopically identical, I might think that there's no selection for or against the minor parent ancestry. Why would there be? But I can also simulate that admixed population under stabilizing selection and just check what happens, right? And so if I do that in SLIM, I find that, in fact, the minor parent ancestry, on average, is selected against over time in the admixed population. Okay, the minor parent ancestry is purged. The rate at which it's purged is faster if the degree of divergence between the parental populations is larger, as measured by FST. And you see this characteristic pattern where the rate of purging of the minor parent ancestry is extremely fast in the first few generations after admixture, and then slows down to a sort of steady long-term decline. Okay, so we're going to try to develop some theory to explain why the minor parent ancestry is purged and why the trajectories of this purging have these features. Okay, so let's return to the simple model. Okay, the first thing we need to know is what the admixed population looks like from a macroscopic perspective. And it's been known for a long time that under this scenario, the admixed population shows increased genetic variance relative to the parent populations. This is known as transgressive variance or segregation variance. Okay, and if we look sort of under the hood at what genomes look like in the admixed population, well, recombination recombines the two ancestries together in these hybrids down the generations and forms these mosaic genomes composed partly of population 1 ancestry and partly of population 2 ancestry. And so we can get various kinds of these mosaics. For the purposes of my argument, I'm going to consider a very simple one, where this haploid hybrid genome has inherited yellow ancestry to the left and blue ancestry to the right. And I'm going to consider exactly the same partition of the genome in the ancestral populations, okay, the two parent populations. So this is population 1. We know that the mean genetic value of a haploid genome in population 1 was 0 because it was under stabilizing selection for that value. So let's suppose the left-hand side of the genome contributed a value of +1 on average to the trait, in which case the right-hand side of the genome must contribute a value of -1 to the trait, right? So that the overall value on average is 0. Population 2 in their period of divergence might have come to a different genomic arrangement, right? It might be that the left-hand side of their genome contributes an average -1 to the trait, and the right-hand side contributes +1 to the trait. I'm just going to assume that that's possible for now. Okay. In that case, both of the parent populations have mean genetic values equal to their optima, but the hybrid genome, you can see, does not. The left-hand side of its genome inherited from the yellow ancestry has a value of -1. The right-hand side has a value of -1. And so because these two regions did not obviously mutually co-adapt in the separate populations during their period of divergence, this hybrid has a suboptimal phenotype. Of course, I could have done it exactly the other way around, right? This hybrid has a value of -2, but if I flipped the ancestries, it would have a value of +2. So by symmetry on average, the hybrids have a mean phenotype of 0, the optimum, but they show much greater variance around that mean than the parent populations do. And this is an effect we can quantify. Okay, so if we look at a gamete that has a proportion of yellow ancestry P, then we can calculate its fitness reduction on average as a function of P, the proportion of the two ancestries, the strength of selection, 1 over VS. Remember, VS is the width of the stabilizing selection fitness function. And this parameter VSEG, which is the segregation variance that was latent between the two parent populations. So VSEG depends on the degree of divergence between the two populations and on the overall amount of genetic variance that was present in the two populations. And so this segregation variance was first calculated by Landi for F2 hybrids, and Yuval and I have extended those calculations to hybrid gametes of any ancestry fractions. Okay, so we now know the fitness reduction of a hybrid gamete. Let's go back to these trajectories and let's see if so we now have some reason that hybrid gametes and hybrids in general have reduced fitness. Can we use that to explain these trajectories? Okay, so let's put down the reduction in fitness of a gamete with ancestry fraction P. Now, if we know in the admixed population, in a given generation after admixture, the full distribution of ancestry fractions P in the population, then we should be able to integrate this fitness reduction against the distribution of ancestry fractions to calculate in that generation the expected change in ancestry fractions. Okay, and that's something we can do. So an individual T generations after the admixture pulse produces a gamete. That individual has 2 parents, 4 grandparents, and so on. And so T generations after the original admixing populations, they'll have 2 to the power T distinct ancestors in those admixing populations, provided T is not too large. Okay. Among the original admixing population, a fraction theta came from the minor parent ancestry and 1 minus theta from the major parent ancestry. So the number among the 2 to the T ancestors of this individual that come from the minor parent ancestry is simply binomially distributed with probability theta. Okay, and if that individual has K ancestors from the minor parent ancestry in the admixing populations, then assuming that all those are unlinked and some other assumptions, the ancestry fraction in the gamete produced by that individual is simply K divided by 2 to the power T. So that gives us the distribution of ancestry fractions among gametes in generation T. We integrate their fitnesses against this distribution, and we obtain an expression for the expected change in the minor parent ancestry in generation T as a function of the time since admixture, the degree of segregation variance, and the fractions in which the populations mix. So can we use this expression to understand the trajectories that we found in our simulations? Okay, well, does the expression show that the minor parent ancestry should be purged? Yes. Theta, the minor parent ancestry fraction, by definition is less than 1/2, and so that term in green over there is negative. We expect the minor parent ancestry to decline over time. We also expect faster purging if the degree of divergence between the parent populations is larger, because the segregation variance depends positively on FST. And we can also recover this pattern where the rate of purging is more rapid in the early generations in this first term in the expression. And I should say that this pattern where introgressed ancestry, by whatever mechanism it is purged far more rapidly in the early generations than the later generations, is very general to models of selection against introgression. And it's a result of the fact that in the early generations after admixture, the introgressed alleles are contained in very long linkage blocks that have not yet been broken down by recombination. And those long linkage blocks have a large combined effect. And if that effect is deleterious, the selection against the introgressed alleles that they contain is therefore much stronger. Okay, so Yuval and I have extended those calculations in a bunch of ways in our preprint. But the general takeaway, I think, is that stabilizing selection on any quantitative trait at all, I mean, say height, in principle is a mechanism of selection against introgressed ancestry. Okay, it's clearly distinct from other mechanisms that have been proposed. So, for example, it's distinct from ecological incompatibilities, because the model that I showed you assumed that the trait optima for the two populations, the two parent populations, was the same. There was no difference in the ecological context. It's different from load-based arguments, because in this model, I assumed the parent populations were demographically identical. Neither had a smaller effective population size. In a way, it's similar to Dobzhansky-Miller incompatibilities, right? Because we think of this region of the genome inherited from the yellow ancestry as being somehow maladapted to the rest of the genome inherited from the blue ancestry. But that also highlights a little bit of a difference from the traditional Dobzhansky-Miller incompatibility picture, where the two substituted alleles are incompatible together, but the ancestral alleles that they substituted from, which were present in the ancestral population, are not incompatible. In this case, yellow ancestry in this portion is maladapted to blue ancestry in this portion. That means that blue ancestry in this portion is also maladapted to yellow ancestry in this portion. Okay, and so you get a kind of symmetric incompatibility from stabilizing selection. I showed you results where only a single trait was under selection. It's pretty clear that if more traits are under stabilizing selection, as we expect many to be, that'll lead to a stronger barrier to gene flow. And a point that I'm particularly interested in, the results I showed you assume that all loci were unlinked. Okay, but we can also just carry out our simulations with realistic linkage maps, and we can see that they have a really large effect. So in these two simulations, I've used the human linkage map and the Drosophila melanogaster linkage map. I've changed nothing else in the simulations. And you can see that Drosophila melanogaster, which is a low recombination species with very few chromosomes, no crossing over in males, shows a much more profound purging of introgressed ancestry under stabilizing selection than humans do. Okay, so clearly linkage plays an important role in this. Now, if I go back to the sort of intuitive explanation of what was going on here, Surya.
Are there new mutations that arise since the population split, or are these simply ancient mutations?
Yeah, no, that's a good question. So the scenario we rarely have in mind is a situation where the two populations are only recently diverged. So they share most of their genetic variants, and they don't have that many private variants. The mathematics doesn't depend on that. What it'll turn out to depend on is just that there be persistent genetic variants in the two parent populations for the trait. It doesn't matter if that genetic variance overlaps between the two populations or not. So it turns out not to depend on that at all. But it's certainly the scenario we have in mind is one where most of the polymorphisms are shared between the two populations, but it doesn't rely on that. Okay, and I'll come to that point in fact in a second. So, right, the basic scenario that we had, we imagined that population 1 from the ancestral state got a value of +1 on the left and -1 on the right to compensate. And population 2 came to a different arrangement with -1 on the left and +1 on the right. Now, we've assumed that that's possible and that it's measured by some degree of FST, but it's not clear that that is possible under stabilizing selection. And I should say, we're not the first to realize that a scenario like that is a bad thing for hybrids. But usually when people are thinking of this scenario under stabilizing selection, they're imagining a situation where an allele substituted on the left-hand side in population 1, a trait increasing allele, and thereafter to compensate for that trait increasing allele, a trait decreasing allele substituted on the right-hand side. And similarly in population 2, maybe a trait decreasing allele substituted on the left-hand side and a trait increasing allele substituted on the right-hand side to compensate. And you can see in that scenario that at least some of those substitutions must be against the grain of selection, right? If they're returning us to the optimal trait value, then at least one must have sent us away from the optimal trait value. So that process is kind of limited by the efficiency of selection, by things like population size, and that's going to limit the rate at which fitness can decline in the hybrids. The scenario that we're imagining is that there's persistent genetic variation in all of these genomic segments in both of the parent populations. But we have, for the time being, simply assumed that given persistent genetic variation, you can get these differences in the mean genetic trait values of the different regions. Okay, but we've simply predicated our argument on that without showing. Okay. And so Pavitra Muralidhar and I have tried to now characterize this process as a general theory. So I'm going to turn to that now. I'm going to imagine again a partition of the genome into, in this case, two segments. I'm going to make it a simple one where there's a segment on the left, a segment on the right. It doesn't depend on that. I'm going to call the segment on the left our focal segment. I'm going to think of it perhaps as just a chromosome. That's chromosome 1, and the rest of the genome is all the other chromosomes. I'm going to say that the mean genetic value of our focal region or our focal chromosome is x of t at time t, and the mean genetic value of the rest of the genome is y of t at time t, so that the genome-wide mean genetic value, z of t, is given just by the sum of x and y. And I'm going to imagine that this trait is under stabilizing selection. I'm going to assume that the focal chromosome carries a fraction L of the total genetic variance, Vg. And given that setup and stabilizing selection on the overall value, we can write down a stochastic differential equation that governs how the mean genetic value of our focal chromosome behaves over time. Okay? It's partly governed by selection on the trait. So I'm going to assume that the optimal trait value is 0. That means that if the trait value goes above 0, our focal chromosome is under selection to bring the trait value down. The intensity of that selection is given by 1 over Vs, and our focal chromosome is going to respond to that selection in proportion to its genetic variance, which is L times Vg. Okay, there's also a random drift term to these dynamics. Okay, so this Xi1 is a white noise process, and overall that term gives the drift in the value of our focal chromosome as a function of its genetic variance. And that drift is, of course, going to be slower if the population size, NE, is larger. So I can write down exactly the same, or the analogous equation for the rest of the genome. Okay, it's under exactly the same kind of selection on the overall phenotypic value, Z of t. It'll respond to that selection in proportion to its genetic variance, 1 minus L times Vg, and it has an independent drift term given by this term on the far right. Okay, so we have a coupled system of stochastic differential equations. One thing we can do with those is simply sum them together. That will give us a stochastic differential equation for the overall mean genetic value. Okay, and what we find is that the overall mean genetic value behaves according to an Ornstein-Uhlenbeck process. Right, that is a random process with a tendency to return to a particular point, in this case, the optimal value of 0. And that's reassuring because it's been known for a long time that a heritable trait under stabilizing selection, its mean genetic value behaves according to an Ornstein-Uhlenbeck process. So that's kind of like a sanity check on the equations that we've written down. But we're not really interested in the mean genetic value genome-wide, right? We know that its behavior is boring. It goes to the optimum and it stays at the optimum. Okay, what we really want to do is characterize the behavior of our chromosome of interest. So what we want to kind of do is decouple these two stochastic differential equations to isolate the behavior of the genomic region of interest. So to do that, I'm going to divide each of these equations by their proportion of the genetic variance to get differential equations for a kind of scaled mean genetic value of the two regions. You'll notice that in these scaled equations, the selection terms are identical. So if I subtract the one equation from the other, I lose those selection terms. Okay, and I find that the time behavior of this thing in square brackets, and I don't really know what that is right now, right, is a simple Brownian motion. It's just a white noise scaled by a certain variance. Okay. I'm now going to make an approximation. So I know that eventually the genome-wide mean genetic value must be close to its optimum and must stay close to its optimum. But that means that the value of the rest of the genome must approximately be the negative of the value of our region of interest, right? And so if I substitute that approximation into the thing in square brackets, I now obtain a differential equation for the mean genetic value of our region of interest. Okay, and if I neaten that up a little bit, I find that the time behavior of the genetic mean of our region of interest, our chromosomes say, is a simple Brownian motion. Okay? So despite the fact that the genome-wide genetic value behaves according to an Ornstein-Uhlenbeck process and is therefore constrained to eventually be close to its optimal value of 0, the genetic mean values of individual subregions of the genome, like chromosomes, are free to drift. Okay, so long as, of course, they compensate for each other. This is something we can simulate. So in these simulations, there's a trait under stabilizing selection. The genome-wide genetic mean is given by the black lines, and you can see that it's constrained to remain near the optimum of 0. But the lines in color are the genetic mean values of a region of the genome that's about 30% of the genetic variance. And so across replicates, you can see that this region can take on substantially different values. The fact that it behaves according to a Brownian motion means that we can characterize the variance across these evolutionary replicates. And we find that the variance in the mean value taken by the region should increase linearly in time. We can decompose that into the rate that we would expect if there were absolutely no selection on the trait. That would be this term LVG over NE times T. So we can see that even though qualitatively the region drifts as if it's neutral, in fact, the rate of that drift is slightly slowed down because of stabilizing selection by a factor that depends on the fraction of the genetic variance accounted for by that region. And that makes sense. If the region's accounting for almost all of the trait variance, then it should be subject to stabilizing selection, and it shouldn't drift that rapidly. But if, for example, it's 1/2 of the genome, then it drifts at about half the neutral rate. Okay, and this is something that we can also check in our simulations. Right, so we find across replicates of our simulations that the variance of a region of the genome that's about 1/3 of it increases linearly over time as if it's a drift process, but the rate of increase is slightly slower than we would expect if there were no selection at all. Okay, so we know that genomic subregions can drift almost as if neutrally. What does that have to say about our speciation example? Okay, so let's go back to this example and imagine that the ancestral population had a value, we can just code it this way, as 0 on the left-hand side and 0 on the right-hand side. We now know that in the period of divergence between the two descendant populations, the region on the left is allowed to drift as if neutrally, right? And so it's independent in the two populations, and they will take on values delta-1 and delta-2. Which means that the right-hand side of their genomes must take on values minus delta-1 and minus delta-2 to compensate for that. If we now form the hybrid genome, we get that it has a phenotype of delta-1 minus delta-2, which need not be equal to 0, right? Because these two regions were not necessarily co-adapted between the two species. And so we can calculate the expected fitness reduction of this individual with phenotype delta-1 minus delta-2. And we find that the reduction in fitness to the hybrid is increasing in time according to the parameters of our model, okay? But you might remember that we had already an expression for the reduction in the fitness of an individual with hybrid fraction P, which in this case is just L, okay? So if we put that expression back up, right, it was in terms of the assumed degree of divergence at trait affecting loci, FST. These two expressions should be equal, but that means that we can obtain an expression for FST between the two populations under stabilizing selection, and we find that it's equal to the neutral expectation. So FST accumulates as if neutrally, even though, and this is at trait affecting loci, even though the trait is under stabilizing selection. And that's a result that, in fact, previously been obtained in a different way by Monty Slatkin and Russ Landy. Okay, you might object because the prediction here is that chromosomes drift in their genetic values for some trait as a Brownian motion. That means given enough time, and that prediction, I should say, depends on position for in these regions for the trait in question. That predicts that over time, these regions can drift, essentially infinitely far from their ancestral values and infinitely far from each other's values. And that seems unrealistic, right? We don't imagine that chromosome 1 in humans, encodes a value of plus 5 million centimeters for height, but luckily chromosome 2 encodes a value of minus 5 million centimeters for height. That seems unrealistic. And there is, in fact, a process that checks this, and that's mutation, right? So a chromosome that has achieved an extremely high value for some trait by virtue of having substituted many trait-increasing alleles, or bringing many trait-increasing alleles to high frequency, right? Mutation will have a greater opportunity to turn those trait-increasing alleles into trait-decreasing alleles than the reverse. And so mutation will tend to decrease the genetic value of that chromosome. Similarly, a chromosome that's achieved a very low trait value because it has substituted many trait-decreasing alleles will give mutation more opportunity to turn those trait-decreasing alleles back into trait-increasing alleles than vice versa. It turns out that if we assume that there's a symmetric per locus mutation rate of mu, this effect is easy to quantify. A chromosome that has achieved value X will have a force by mutation reducing its value by -2 times mu times X. So if X is positive, it will tend to be decreased by mutation. If X is negative, it will tend to be increased by mutation. Okay, and so we can just add those back into our stochastic differential equations, go through exactly the same procedure as before. And what we find now is that the behavior of the mean genetic value of our region of interest is in fact an Ornstein-Uhlenbeck process. So no longer a Brownian motion, but an Ornstein-Uhlenbeck process. However, it's an Ornstein-Uhlenbeck process with an extremely small selection term, a selection term that depends on the per locus mutation rate. So the picture that emerges here is that in the short term, the behavior of this, the genetic value of this region looks as if it's a Brownian motion. But in the long term, it can't go too far away from zero in either direction. And what it actually looks like is a random process that traverses an enormously wide normal distribution. So it still has enormous latitude to drift around relative to the genome-wide genetic value, but it doesn't have infinite latitude. Okay, so what I've shown you here in this theory is that the chromosome scale dynamics under stabilizing selection, this kind of chromosome level drift, is qualitatively distinct from the genome-wide dynamics, right, which are simply that the genome-wide value moves to the optimum and stays there. And they're also distinct from these kind of underdominant-like allele frequency dynamics at individual loci. So we've identified a kind of qualitatively different scale of the dynamics under stabilizing selection. It looks like random drift in the short run, but in the very long run, there's some kind of constraint induced by mutation. And something I haven't showed you, but is going to be important for what I talk about next, is that these dynamics, this sort of chromosome scale drift, is not slowed by pleiotropy, right? So it might be that a mutation that affects a particular trait under stabilizing selection also affects some other trait under stabilizing selection. And you might think, therefore, that selection on those two traits constrains the kinds of dynamics that are achieved. And it turns out actually, nonetheless, a region, the genetic value of a region for a particular trait remains a Brownian motion even in the presence of pleiotropy. Okay, so with that, I'm going to talk about the second application that I wanted to discuss, a second evolutionary application. And this has to do with evolutionary transitions in sex-determining systems. Okay, and as I said, this is work of my colleague and collaborator, Pavitra Muralidhar. If you look across the sexual tree of life, you see an enormous diversity in the mechanisms by which sex is determined. Okay, but if you look closely, you will notice that by far the most common mechanism by which sex is determined is genetically, by the presence of sex chromosomes. And these sex chromosome systems have a bunch of different flavors. The most basic is whether the female carries the sex-specific chromosome, as say in birds, or the male carries the sex-specific chromosome, as in mammals. Another difference between these systems is whether the sex-specific chromosome, say the Y, is degenerated relative to the X chromosome, or whether the two sex chromosomes look the same, either cytologically or in terms of their gene content. Another possibility is that a clade might show the same sex chromosome system writ large, but the linkage group that serves as the sex chromosomes differs from species to species, as in these frogs. Okay, so again, looking at this phylogeny from the tree of sex database, you'll see that there must have been evolutionary transitions between these sex chromosome systems, right? There must have been turnovers in the sex-determining mechanism. And with respect to sex chromosomes, these can take a number of forms. Okay, one form of a transition is that we switch the system of heterogamety, say from male heterogamety to female heterogamety, but the linkage group that serves as the sex chromosomes remains the same. Another possibility is that we maintain in the transition the same system of heterogamety, here male, but we switch the linkage group that serves as the sex chromosomes. Or we could do both. We can switch the system of heterogamety from male to female heterogamety here, and switch the linkage group that serves as the sex chromosomes. So all of these transitions are possible, and a lot of theory has been done on the mechanisms that might generate these transitions, right? It turns out that theoretically they should be quite simple. And there are lots and lots of things that should drive them. But if we go back to this phylogeny, another thing we might notice is that there's actually quite a lot of stasis of sex-determining mechanisms in this tree. I mean, this is a tree across an enormous swath of life, and we look within these very broad clades, we actually see quite a lot of stasis. And so maybe a more salient question is what explains this stasis? Why do sex chromosome systems remain remarkably stable over time, given a theoretical expectation that they should actually be quite labile? Okay, well, to study this question, let's focus on a particular sex chromosome transition, right, as has in fact happened in this Medaka fish. So this is a transition in the system of heterogamety that also changes the linkage group that serves as the sex chromosomes. Okay, so how might this happen? Well, let's look at the original system of male heterogamety. Okay, so we have XX females and XY males. And we imagine that a mutation appears in an autosome that is dominant female determining. Okay, so an individual carrying this mutation, even if they carry a Y chromosome, will develop as female. Okay, so that genotype that I've just put there is a female genotype. If we now carry out all the various possible matings between the male and female genotypes and look at the progeny genotypes that result, we get a closed system of six sexual genotypes. And you'll notice that the bottom pair of genotypes defines a ZWZZ female heterogametic system, right? With the previous autosome now serves as a sex chromosome, and the previous sex chromosome has reverted to being an autosome. So the question that Pavitra is asking is what stops these transitions from happening? And there are some relatively simple explanations in certain cases, right? One case is where we're looking at a particularly old sex chromosome system where the Y chromosome has had a long time to degenerate. In that case, presumably these individuals that are homozygous for the Y chromosome and don't carry this important X chromosome are not going to do well. And their reduced fitness is going to prevent that transition from happening. Another possibility is that the sex chromosomes, with their strange sort of sex-biased transmission dynamics, might carry sexually antagonistic alleles, right? And there's been a lot of theory arguing that they should preferentially carry sexually antagonistic alleles, alleles that increase fitness in one sex and decrease fitness in the other. Okay, this is clearest to see for the Y chromosome, which we might expect should have, alleles that increase male fitness. And since the Y chromosome is never present in female, those alleles might decrease female fitness, right? So these females that are produced in the course of this transition and carry a Y chromosome might suffer the adverse effects of those sexually antagonistic alleles on the Y, and that will prevent transition from happening. Okay, these explanations kind of rely on the unique transmission dynamics of sex chromosomes. Okay, and what Pavitra has shown, and you might have guessed this from the title of the talk, is that in fact stabilizing selection on any quantitative trait itself acts as an impediment to sex chromosome turnover. Okay, and so the model that she's considered is very simple. There's a quantitative trait that's under stabilizing selection in both males and females. That's for the same optimal value, turns out not to be important. Importantly, mutations that affect these traits affect both male and female trait values, but not in exactly the same way. So mutational effects on male and female trait values are correlated, but not perfectly. Okay, and that's justified from estimates of effect sizes for a large number of quantitative traits. We assume that the trait is encoded across the entire genome, that is across the autosomes, the X chromosome, and the Y chromosome. We're imagining a system where the sex chromosomes are not that old, and so the Y chromosome has not yet had time to degenerate. And there must be no recombination between at least some part of the X and the Y chromosomes, okay, which is common even in young sex chromosome systems. Well, in that case, we know what happens to the mean phenotype under stabilizing selection in males and females, right? The genome-wide mean remains close to its optimum. But we also know that the individual chromosomes, including the sex chromosomes, can drift over time in their values, so long as they compensate for one another to ensure that the genome-wide mean is close to its optimum. What does this mean for our turnovers? Well, importantly, all turnovers of sex chromosome systems create novel sexual genotypes. In this particular turnover, the novel sexual genotypes are given in colored boxes here, right? So we have an XY female. There were no XY females in the original system. And we have individuals that carry two Y chromosomes. That was not possible in the original system. And importantly, those novel sexual genotypes are going to disrupt the kind of cross-chromosome balancing act that was set up under chromosome-specific drift under stabilizing selection. So they're going to be less fit, and stabilizing selection on them is going to hinder a turnover. Okay, let's focus on this individual just to see exactly how that works. Let's imagine that ancestrally, in males, the X and the Y and the autosomes all had mean genetic values of 0. We know that over time, these values are going to drift, right? Under this process of chromosome-specific drift. So the X might have come to a value of +1, the Y might have come to a value of +2, the diploid set of autosomes to a value of -3. Okay, and together they sum to 0, so the individual is still at the optimal value. But now suddenly, in the course of the transition, you have a novel male genotype with 2 Y chromosomes, and that male finds that his genetic value is now set away from the optimum. So that individual's disfavored, and that's going to hinder the sex chromosome turnover. How much is it going to hinder the sex chromosome turnover? Well, Pavitra's carried out simulations of this process under reasonable parameter regimes, and she finds that in these regimes, over time, this chromosome scale drift causes the fitnesses of these novel sexual genotypes to decline pretty substantially on realistic timescales. Okay, so this is going to, early on not be such an impediment to sex chromosome turnover, but much later, because the fitnesses of these genotypes are so low, it's going to be a major impediment to sex chromosome turnover. Again, and I've highlighted this for a particular case of a transition. It turns out that it's true for absolutely all sex chromosome turnovers. So a turnover that maintains the system of heterogamety but changes the linkage group, the system that changes the system of heterogamety but maintains the linkage group. It's even true, it turns out, for a transition between genetic sex determination and environmental sex determination. Right? Under environmental sex determination, all chromosomes are autosomes. And so in that transition, a sex chromosome is lost. So this seems to be a very, very general mechanism preventing sex chromosome turnover, relying only on the existence of stabilizing selection on quantitative traits. Okay, some extensions that Pavitra has drawn, and I encourage you to have a look at the preprint that she's posted on bioRxiv. So importantly, this mechanism does not preclude turnovers entirely, but as I just mentioned, right, it precludes them more and more over time. So the picture is one where either sex chromosome systems turn over rapidly, or if they don't turn over rapidly, they get stuck. And that has implications for phylogenetic patterns of heterogametic systems. And it is, in fact, the observation that you have sort of very mature systems like in mammals and birds, or you have clades where you get repeated rapid transitions. So it seems to be consistent. Obviously, this mechanism is going to be stronger if there are more traits under selection, and selection is stronger. The greater the fraction of genetic variants contributed by the sex chromosome, specifically the non-recombining region, the more important the theory is going to be. And importantly, this also doesn't preclude the other mechanisms that people have worked out that prevent sex chromosome turnover. Okay, so sort of to conclude, I hope that I've shown you that stabilizing selection, despite being superficially the most boring kind of selection, can have some very interesting evolutionary implications. It, for example, generates selection against introgressed ancestry. It impedes sex chromosome turnover. And in both cases, those phenomena turn out to be driven by this novel sort of intermediate scale genetic dynamics that stabilizing selection generates. And just to finish off, I want to say that, we've developed the theory of this chromosome scale drift to explain specific examples that, well, Pavitra and I, in fact, independently came to. But, looking at these equations, it turns out that this theory should be substantially more general, right? We have a component X and a component Y that sum to a component Z under stabilizing selection, right? So in fact, this is kind of a theory of quantitative system drift, the idea that though the overall value produced by some, say, developmental system is constant across time, different species can show different values in the components underlying that system. And so to the extent that this is a general theory of systems drift of quantitative traits, it should have applications that reach beyond those that I've given you today. So for example, we're considering scenarios where species are involved in a mutualism, a public good is produced, there's some optimal level of that public good. Do the contributions of the two species to that public good, like an enzyme or whatever it is that's secreted into the environment, do they drift over time? Genes can be regulated in lots of different ways at the same time. Do we see a similar drift process between these various ways that a gene can be regulated if the overall expression of the gene is under stabilizing selection? And the strange pattern where the rate of drift in genetic value of a region of the genome depends on the region's size suggests potential for cross-population tests for stabilizing selection. Okay, and just to finish off, I'll plug that I've recently started a lab at the University of Chicago, and I'm actively recruiting grad students and postdocs. Thank you.